I'm excited to introduce Peng Zhang, who's a research scientist at Stony Brook University, talking about integrating machine learning and multiscale modeling for blood flow and platelet-mediated thrombosis initiation.
Okay.
And once again, for those of you who click on Peng Zhang's title, and you'll find the comments for him to start posting questions for Peng. And Peng, you are sharing your screen.
Yes, I'm sharing my screen, and hope that you can see that. So, good afternoon, everyone, and thank you for the introduction. It's my pleasure of taking this opportunity to present my research work about integrating machine learning methods into the multiscale modeling framework for the study of platelet-mediated thrombosis initiation. I also thank my team. So first of all, let me introduce the biomedical problem I'm studying. We focus on simulation of the flu-induced platelet activation and aggregation, which promotes the thrombosis formation. How do I study? And we have developed the particle-based multiscale modeling framework, and I show the general picture and in this figure, and to simulate the process of platelet dynamic interactions with the injured vessel walls. So in this process, we focus on simulating the platelet flipping and deforming and approaching to the blood vessels, and also the platelet aggregation, adhesion, and activation. I also put some key results and in the animations. So in this process, we start with the platelet flipping in the free flows, and the platelets are allowed to flip and deform, responding to the applied dynamic shear stresses on the platelet membrane. Also, we have modeled the GP2B3A receptors, and that can bind to the fibrinogen. So the simulated dynamic bonding of the GP2B3A fibrinogen, GP2B3A bridging will initiate and enhance the platelet aggregation under the flow conditions. And as the platelet is approaching to the blood vessel walls, the adhesion of the platelet to the blood vessel wall is initiated through the dynamic bonding and between the JP1B receptor and the collagen. So, and the platelet is also first flipping over the VWF coated blood vessel walls and finally attached to the blood vessel walls. And also we use this model and continue to simulate the recruitment of aggregation of the flowing platelets, and by the activated platelets deposited on the blood vessel wall. So the next question is that, what is my numerical modeling framework to simulate the whole process? Actually, our multi-scale modeling framework is a particle-based methods. It has incorporated 3 simulation methods. The very bottom scale is the molecular dynamics. We use the atoms to simulate the internal structures of the platelets, including the cytoskeleton and the actin filaments. The very top scale method is based on the dissipative particle dynamics (DPD) method to simulate the viscous blood flow and also the blood vessels as the boundary conditions. In the middle, and we developed the CGMD methods and as the interface between the MD and the DPD scales. And we use the CGMD methods to simulate the platelet membrane as the mechanotransduction pathway of the dynamic shear stresses from the surrounding flows and to the internal structures. So next, let's look at the overview of our numerical multi-scale modeling framework. As for most of the modelers, also we are facing 2 challenges. One is the computational cost, the other one is the validation of our predictive in silico data. That is why we have introduced 2 machine learning pipelines and to address these challenges more efficiently. So here, this is the first pipeline used to synthesize the sparse and noise in vitro data from the experiments. The purpose is to establish the numerical correlation across the different spatial and temporal scales between the in vitro experiments and also the in silico simulations. So first, let's look at the left branch of the figure. We developed the active learning-based systems for the platelet segmentation from the in vitro experiments. The segmented data and together with the biophysical conditions will be input into the second fully connected neural networks to do the predictions. For two things, one is that we want to predict the contact area and attachment between the aggregated platelets. The second thing is that we want to predict the platelet adhesion events, including the flipping and reattachment periods. On the other hand, we designed the second machine learning methods and to speed up the simulation process by adapting time step sizes to the underlying biophysical phenomena during the simulation. So this is an AI-guided adaptive time stepping ATS method. So conceptually, the ATS method is kind of cross-screening in the time scales to compare the DPD, CGMD, MD method is kind of cross-screening in the spatial scales. So now in the previous slide, we defined the biomedical problem, and this slide, we defined the methodology for solving the problem. So next, let's briefly look at all of the particle-based governing functions corresponding to different scales. In the molecular dynamics, we use the atoms to simulate the nanoscale structures of the cytoskeleton and plus the actin filaments that is protrusible during the platelet activation. At the top scale, we use the DPD method and to simulate the viscous flow and plus the boundary conditions and inlet outlet boundary conditions. In the middle, we developed the CGMD cross-grained molecular dynamics to simulate two things. One is the membrane, platelet membrane, that can deform responding to the applied shear stresses. And on the other hand, we are also using the CGMD method to simulate the platelet receptor ligand interaction. So here, and using this framework, we have enabled the dynamic interaction of the deformable platelets with the dynamic blood flows. And the platelet is flipping and flowing in the shear flows, and also the dynamic shear stresses is continuous mapped on top of the platelet membranes, transmitted to the platelet cytoplasma systems, and finally mapped onto the cytoskeleton structures. During the simulations, we calculated the accumulated of shear stresses on different components so that we use this data can find out where is the high shear stress locations. And then we use this data as a GAT and to find out where we should trigger the pseudopode formation. And during the platelet aggregations, and we do the same measures for the dynamic shear stress accumulations, and also we will predict the contact events between aggregated platelets. So the next challenge is that how can we validate the numerical predictions? My colleague conducted the in vitro experiments under the same shear stress conditions. The purified platelets passing through the VWF-coded microchannels. As a modeler, my work is started with the streaming data, and the camera is attached to the microscope and produces the high-speed and high-resolution images. The generation speed is 1,000 frames per second, that is to say 1 frame per millisecond. So next, we have designed the active learning frameworks for the platelet segmentations with focus on the platelet boundaries, details, including the pseudopodes and memory tethers. Those images can give us the 2D geometry information for the platelets, and then we build the three-dimensional mathematical model to find out the major and minor axes, thickness of the platelets, and also we will just estimate the rotation angles for those platelets, so that we can find out the three-dimensional shape data and for all of the aggregating platelets, and we save them in the database. Here, let's see some the general structures of our semi-unsupervised learning systems we developed for the platelet segmentation. So this is a kind of active learning. In the first layer, we have 7 agent networks to do the cost range segmentation results. All of the preliminary results together with the raw data will be input into the policy networks and to generate the fine-grained final output. And also in each experiment, we have to handle millions of the segmentation results. So we designed the reward network as the quality control for the segmentation results. So we compare the segmentation results with the raw data and then produce a scaler to show the quality of the segmentation results. For high quality, we directly save them in the database. For some low quality segmentation things, we will just ask human experts and to do the relabeling and ready for the next training period of our machine learning systems. So next, let's look at some prediction results from our active learning systems. So first of all, we can say that the machine learning systems can accurately differentiate the boundaries and for the platelets with pseudopods and the platelets with membrane tethers. And even for some irregular shapes of the platelets, we can also find out the good boundaries. So in the next step, and we use the 2-dimensional information and plus the 3-dimensional mathematical model for the contact angle and distance between aggregated platelets, plus some in vitro setup, for example, the shear stress conditions, input all of the values as now for the fully connected deep learning neural networks. We're training these neural networks to predict the contact areas between the aggregated platelets. And we use the 3 different shear stresses, 1, 5, 10, 10 per centimeter squares to train the networks. To validate the results, actually, we used the totally new shear stresses 6.7 and to test the results. So here, this is the model predictions and experimental results. We can see that the data are perfectly matched. So it means that we can our machine learning model can accurately predict the contact area, and that can be used in our multiscale modeling frameworks to validate our in silico results. Using our simulation systems, we can simulate and compare the flexible models of our model system and with the oversimplified rigid model systems. We can see that if you neglect the platelets deforming capability, you will significantly underestimate the attachment degree and during the platelet recruitment. So, and in the previous slides, I have talked about the machine learning pipeline to optimize the modeling and its experimental validation. So in this slide, I want to talk about the other machine learning pipeline to reduce the computational cost without losing accuracy. Traditionally, and our modelers will do the modeling and optimize the performance and by ourselves. However, my question is that, can we teach the supercomputers to optimize the simulator performance by themselves? Yes, we can. So this is our new algorithm, adaptive time-stepping algorithms, and we we do the online learning and collect the physics measures and do the denoising, and then we use the RNN, recurrent neural networks, to extract different latent features. Then we can predict the best step sizes and step jumps. Step sizes will be feedback to the simulation systems to speed up, and using compared with the standard methods, we can see our ATS method and almost generates the identical results, but we can unlimited more than 40% unnecessary computing. So to summarize, and we have presented two machine learning pipelines, and one is for the to improve the model credibility and extracting the in vitro platelet geometries from the experiments, and then using the deep learning networks to synthesize the platelet geometries. On the other hand, we use the online learning systems and to adapt the time step sizes to eliminate unnecessary computing steps while maintaining the enough accuracy. So last, I would like to thank my colleagues from the Departments of Biomedical Engineering and Applied Math at Stony Brook University, and thank all of the funding resources, and also especially thank my mentors, Professor Danny Bluestein and Professor Yuefan Deng. So thank you so much.